In this lecture, we're going to talk about the link reaction and Krebs cycle. So with respiration, we have already spoken about glycolysis. If you haven't watched that mini lecture now, go back and watch that one first. So this bottom part of the graph here, this image here is aerobic respiration. So respiration in the presence of oxygen. And what we're going to focus on today is the link reaction and the Krebs cycle. So it's this part that occurs in the presence of oxygen. So in the link reaction and Krebs cycle, you won't see oxygen being used up. But if oxygen isn't present, they won't occur. And they will take the pyruvate, the product of glycolysis, and break it down further. Glycolysis occurs in the cytoplasm of the cell, and with the following stages in respiration, they all move into the mitochondria. So link reaction and the Krebs cycle take place in the matrix of the mitochondria, and then the final stage in aerobic respiration, the electron transport chain, that will take place on the cristae in the mitochondria across the inner membrane. And we're focusing on the link reaction and Krebs cycle today. So we're in the matrix and the mitochondria. And it's worth looking at the structure of the mitochondria again, just to remind ourselves and refresh our memories with mitochondria. So they're there for aerobic respiration. So that's what we're talking about now. They have their own DNA and their own ribosomes. This means they make their own proteins. And it means that in the matrix itself, so if the matrix is here, and in the matrix, this is where we're going to do link reaction and the Krebs cycle, it means that the enzymes needed to do the link reaction and Krebs cycle, the enzymes only have to be here. So it means it's more energy efficient. You don't have to produce all these enzymes and have them throughout the entire cytoplasm of the cell. Instead, you can concentrate them into the small space inside the matrix. So it's a much more efficient system for doing this. With the mitochondria, the other parts that we need to remember, this inner membrane here, the little cristae, the little folds that occur, this is where the final stage, the electron transport, takes place. So it's got the electron transport chain. So lots of special carriers made of protein embedded in the membrane. And also we've got an enzyme called ATP synthase embedded in this membrane. Again, we'll talk about this in our final aerobic respiration mini lecture, but they are in that membrane itself. And remember, we've talked about how the cristae themselves have a very large surface area, and this very large surface area is so that we have lots of electron transport chain carriers and lots of the ATP synthase embedded there, so we can get a very efficient process. The outer membrane of the mitochondria has carrier proteins, pumps that allow molecules to enter into the mitochondria. And for today's purposes, this is where they have pyruvate pumps, so that the pyruvate that was made in glycolysis in the cytoplasm can be pumped by active transport into the matrix so that we can do the link reaction and the Krebs cycle. The last important part to mention is this intermembrane space. So it's the gap between, so if I draw a little cartoon, it's the gap between the outer membrane and the inner membrane. And this intermembrane space is a small space, and it allows us to build up a large proton gradient, which will be really important in the electron transport chain for generating lots of lovely ATP. So we'll just have a very quick review of glycolysis. So remember, in glycolysis, we start with glucose, so a 6-carbon compound, and we're going to break it down to 2 3-carbon compounds. So we get 2 of these per 1 original glucose. Each pyruvate has 3 carbons in it. like we said, it occurs in the cytoplasm of the cell, and the products it makes, we have the pyruvate. So we have 2 molecules of pyruvate as shown in the yellow highlighter. We have 2 molecules, oops, wrong pen, 2 molecules of reduced NAD. Remember, we can also call that NADH. And these 2 molecules of reduced NAD will feed into the final stage of aerobic respiration in the electron transport chain. And the last thing that we have produced is ATP. Now remember, we talked about how the initial stages of glycolysis use 2 molecules of ATP, and the later stages produce 4 molecules of ATP. So the net amount of ATP is 4 minus 2, which is 2 molecules of ATP. You can see the link reaction summarized in the image shown here. And again, remember, we're just looking at an overview of these processes. We're not learning the name of every single metabolite and every single enzyme that is catalyzing this. So we start with pyruvate, and we said that pyruvate will enter into the mitochondria matrix by carriers or pumps, by active transport, and then it will undergo a process that we can call oxidative carboxylation or oxidative decarboxylation. So we are going to remove a carbon dioxide. So we've got 3 carbons in pyruvate at the end of the link reaction. From the original glucose, we've got a 2 carbon group called an acetyl group, like this. In the process of producing this 2 carbon compound, we're also going to produce a molecule of reduced NAD, and remember, we can call that NADH if that is easier for you. So it's used to make that reduced NADH. The acetyl group is a 2 carbon compound, and it joins with a coenzyme called coenzyme A, which helps deliver the acetyl group into the Krebs cycle. So the acetyl group binds with coenzyme A and forms acetyl coenzyme A. Or you could just call it acetyl CoA if it's easier. So you could just call it acetyl CoA, and acetyl CoA is delivered into the Krebs cycle. Now we need to remember that at the beginning we had a glucose molecule, which was 6 carbon, and at the end of glycolysis we have 2 molecules of pyruvate, which are 3 carbon. So that means everything here happens twice per original glucose. So we'll have 2 pyruvates producing 2 molecules of carbon dioxide, 2 molecules of reduced NAD, and 2 molecules of acetyl joining to form 2 molecules of acetyl coenzyme A. So it's worth mentioning twice per original glucose. And we can summarize the reactants and products of the link reaction in a table like this. Now again, it's really important to mention this is for each original glucose. Okay, so for each original glucose, the link reaction happens twice. So we had 2 3 carbon pyruvate molecules, and they will be converted to 2 acetyl CoA groups that join to coenzyme A, and in the process the pyruvate is decarboxylated, so a carbon is removed in the form carbon dioxide. So we have 2 molecules of carbon dioxide produced per original glucose. And we produce 2 molecules of reduced NAD, which will feed into the electron transport chain in our next mini lecture and produce lots of lovely ATP. We then have the Krebs cycle. So still in the mitochondrial matrix, so same location, and you've got all the enzymes for the Krebs cycle in the right place. And what we will do in the Krebs cycle is we will take the acetyl group that is delivered by coenzyme A into the Krebs cycle, and we will completely oxidize it and break it down to carbon dioxide. So at the end of the Krebs cycle, all the 6 carbons that were in the original glucose molecule will be broken down to carbon dioxide. So we will be again doing some decarboxylation. Remember, D means removal as well. In the process as well, we will do some more dehydrogenation, so removal of hydrogen, and that will be used to produce reduced NAD and also reduced FAD. Remember, we talked about those 2 molecules that are involved in these redox reactions here. Krebs cycle only occurs when oxygen is present. So we class it as aerobic, even though you won't see oxygen actually being used in the reactions. But if oxygen isn't present, you get this kind of feedback system where you can't do any of these processes, so you can't do the link reaction and the Krebs cycle. In addition to the decarboxylation and dehydrogenation, we're also going to make some more ATP as well, directly. So we said when you make ATP directly, it's called substrate level phosphorylation. And so in the Krebs cycle, we will have some more substrate level phosphorylation occurring. Now again, we are very much looking at an overview of this system. We are not learning all the original details in vast amount of detail. It's enough to look at an overview. And we can see what's happening in this overview picture here. So I'm going to highlight the different parts as we go through this, just to make it very, very clear. So the top part, the pyruvate to acetyl coenzyme A, remember this is the link reaction. So we've still got the link reaction here as well. And what's going to happen is the acetyl coenzyme A is going to come and deliver the acetyl group in. The acetyl group joins to a 4-carbon compound. So 4 carbons in the 4-carbon compound, 2 molecules in the acetyl group, and you form a 6-carbon compound called citrate. So it forms citrate. And then the Krebs cycle is a cycle because all the stages that follow this are all stages to go back and remake the original 4-carbon compound right at the start. This 4-carbon compound is called oxaloacetate. So you can probably remember that word name. Just remember a few names. So you've got this oxaloacetate here. So the 4-carbon oxaloacetate combines to the acetyl part of the acetyl coenzyme A, and you form the 6-carbon compound called citrate or citric acid, whichever way you want to refer to it. And so we are going to do some decarboxylation to get back to 4 carbons. So I'm going to color code the decarboxylation, and I'm going to color code that. Not that color, that's a horrible color. Let me try that again. So we are going to have our decarboxylation in green. Okay, so we are taking the 6 carbons in the citric acid or the citrate, remove a carbon, and then you've got a 5-carbon compound that you don't need to remember the name of. And then we'll decarboxylate again, and then go through a series of different reactions, which we don't need to remember the details, and we'll go back to the original 4-carbon compound we have. So we're producing 2 molecules of carbon dioxide per acetyl-CoA that is involved. Remember, this all happens twice because you had 2 molecules of pyruvate, therefore 2 molecules of acetyl, join into the CoA to be delivered into the cycle. So decarboxylation is in green. In yellow, we are going to have the dehydrogenation. Horrible word. So we're going to have the dehydrogenation in yellow. Remember, D means removal. So where hydrogen is removed. So we have some reduced NAD made, some more reduced NAD made, and another batch of reduced NAD made. So 3 molecules of reduced NAD for one go around the Krebs cycle. We also produce 1 molecule of reduced FAD. But again, as it all happens twice, that will be overall 2 molecules of reduced FAD. I also said that we do have some substrate level ATP production. So that's shown down here. So this is substrate level phosphorylation producing ATP directly. But with the Krebs cycle, the cycle is because we go back to the original 4-carbon compound to keep the cycle going. At the end of the Krebs cycle, all the carbon that was in the original glucose molecule has been completely broken down to carbon dioxide. And we have to remember that this happens twice because you started off with a 6-carbon compound in glucose, and that breaks down to 2 acetyl groups, which have 2 carbons each. So if we just look at the reactants and products of the Krebs cycle, we're not balancing chemical equations here beautifully like you would do in chemistry, but rather looking at an overall description of what is happening. So you have 2 carbons in this acetyl group, and at the end of the Krebs cycle, this will be completely broken down to 4 molecules of carbon dioxide, and we will have 2 molecules of coenzyme A that are recycled back to keep the link reaction going. Remember, coenzymes are needed for reactions to occur, but just like an enzyme, they're not used up in the reaction. We will also produce 6 molecules of reduced NAD and 2 molecules of reduced FAD. And remember, this is per glucose molecule to begin with from right at the beginning. And we also produce 2 molecules of ATP directly, and this is the substrate level phosphorylation. The last thing to mention is we're looking and talking here about the breakdown of glucose, but we know from the food we eat, we need to digest different materials. So fatty acids, for example, they can be digested and turned to acetates that can enter into the Krebs cycle in the same way, so they can join in as acetyl groups. Amino acids, so when you're breaking down proteins, what will happen to them? They will have their amino group removed, so they'll be deaminated, and the remaining group can then, depending on which amino acid it is, will either enter into the Krebs cycle or enter into the link reaction and then the Krebs cycle. In our next lecture, we will talk more about the electron transport chain. Thank you.